Hello everybody, welcome back to another video, Biology, Cells, and Tissues. Today we're jumping into neuron function. As always, leave a like on the video as it really helps out. Subscribe if you haven't, and let's jump right in. So with neuron function, we're going to talk about something called resting membrane potential, as well as action potentials. We're going to talk about sodium and potassium ion channels, as well as pumps. And we are going to talk about some of the events with generation and propagation of action potentials. So this material, kind of like a couple of other things we've discussed, is going to come back with what I would call with a vengeance. Physiology 1, which is a Try 2 course, one of our first kind of that first week of stuff, you'll talk about membrane physiology, membrane potentials, all sorts of stuff with that. And all of this information is going to come back. With that being said, actually it's going to come back and you're going to go a little bit deeper into it. With that being said, I went ahead and I actually took some of the information from Physiology 1 that I feel like is going to be better at giving us a little bit more of a foundation, and I put it into this PowerPoint. So some of this information is going to be extra for what you need to know for the exam in this specific course, but understand that it's going to come back, and I want to make sure that you guys have a solid understanding of this to make next trimester a little bit more manageable. So cell membranes. This is something coming from Physiology 1. And a lot of information that we should already know at this point. We have our phospholipid bilayer of all of our membranes, and there's a bunch of proteins and carbohydrates stuck all throughout it. Specifically, there are some proteins that are called transmembrane proteins. Sometimes these are called intermembrane proteins. But the technically correct word for these are transmembrane proteins. Okay. The transmembrane proteins, all that means, I have a lot of words in here. You can pause and you can read through this if you would like. But essentially what that means is that the protein goes through the entire membrane. Part of it sticks out the top, part of it sticks out the bottom. You can kind of compare these. I'm not sure if we've quite reached that yet in biochemistry, but if you, I think it's the last week of biochemistry, you talk about the electron transport chain, and you have a couple of these protein complexes. Complex 1, 3, and 4 are all transmembrane proteins, meaning that they go all the way through the membrane. And specifically, the ion channels are designed to allow ions like potassium, like sodium. Other ones that we'll talk about are calcium and chloride. They allow these ions to travel through the membrane, where in other ways, without these ion channels, they would never be able to make it through the membranes. So, ion channels, so there's specifically two that we're going to focus on here. Ligand-gated or chemical-gated, and voltage-gated ion channels. There are other ion channels out there that we will see pop up, not in this trimester, but next trimester. But we will get to those, when we get to those. These are the two that we want to focus on for this. And we're going to take a little sidestep and jump into the nerve stuff, and we're going to come back to those ion channels. So just wait for those for a little bit. So in general, when we talk about our neurons and the neuron function, the whole job of neurons is to send nerve impulses. These impulses, these electrical signals, travel through all of the nerves, and that's how our body communicates with everything. That's how we feel things, it's how we see things, it's how we smell things, it's how we move, how our brain processes, how we think. All of that is through these electrical signals that run through the nerves. We have a couple of words that I want to make sure we know the definitions of. The first is excitability. So this is the ability of those specific cells, of our nerve cells, to respond to some kind of stimulus by generating and then passing along or propagating electrical signals known as action potentials. And those action potentials are rapid changes in the voltage or the membrane potential that is across a cell membrane. We're going to talk more about what the membrane potential of that is in just a second. So our excitability is cells that can send signals. That's what excitability means. If a cell is excitable, it can send signals. The action potential is how it sends those signals. So membrane potential, specifically resting membrane potential, is a difference. Actually, I should say this. Membrane potential is a difference in electrical charge across the cell membrane of a neuron. So what we do is we look at the cell, and almost all of our cells are negative on the inside because our proteins, most of our large proteins, so you think like some of the organelles and a bunch of the proteins that are being made inside, they are highly negative. So the insides of our cells, or the insides of our neurons, are going to be negative, and the outside of them is overwhelmingly positive because we have a whole bunch of ions, especially sodium, on the outside. Resting membrane potential is what we could call the resting or the standard state or the inactive state of a cell, where if it's just kind of sitting there in homeostasis, in equilibrium, everything's balanced, it's just hanging out, not doing anything, it's going to have a specific charge, and that is the resting membrane potential. For neurons, that sits at about negative 70 millivolts. We measure the electrical charge in millivolts, but it can range anywhere from negative 60 all the way down to negative 90, depending on the cells. In Physiology 1, you're going to look at all of this. You're going to look at neurons, you're going to look at smooth muscle, you're going to look at skeletal muscle, and you're going to look at cardiac muscle, and how all of their resting membrane potentials are slightly different. Overall, when we talk about ions, the main two that we focus on are potassium and sodium when we talk about neurons. Potassium is found primarily inside the cell, and sodium is primarily found outside of the cell. Stupid little way that I like to remember this is remember banana in the ocean, because bananas, almost everybody knows that they have lots of potassium. I think the reason why most people know that is because there's a Simpsons episode where Homer says bananas have potassium, or bananas are full of potassium, something like that. But most people tend to know the fact that bananas have potassium. So banana in the ocean. There's lots of potassium on the inside, lots of sodium or salt on the outside. And the resting membrane potential, this balance of potassium inside, sodium outside, and this specific voltage, is all maintained by a special type of transmembrane protein called the sodium-potassium ATPase pump. This is the star of the show when we talk about the cells and all of this membrane potential stuff, because it has one of the most important jobs. The sodium-potassium ATPase pump is an intermembrane or transmembrane protein, and it uses one ATP to move sodium and potassium against their concentration gradients. What that means is there's lots of sodium on the outside, so typically if we were to just leave everything around or leave everything open, sodium wants to come inside of the cell because everything likes to go from high concentration to low concentration. When we have to go against that, we need to use extra energy in order to force that to happen. And that's called active transport. So sodium, potassium, ATPase pump, anything that uses ATP to move stuff, that is going to be active transport. What this pump does is it takes 3 sodium and takes it from the inside of the cell and dumps it to the outside of the cell, and then it grabs 2 potassium from the outside and brings them back inside. It consistently does that to help maintain, as well as to, let's say, restore and maintain the resting membrane potential of the cell. We'll talk about that restore part in just a little bit. So step by step, this is going to be super important for the test. The professors love to test on sodium, potassium, ATPase pump. Because it's so important and plays such a critical role in stuff, they love to ask questions about it. So, the sodium, potassium, ATPase pump starts, I'm going to do start on this one right here. It starts being open towards the inside of the cell. And it invites 3 sodium in. They fit into these slots. And then an ATP donates its phosphate, so it phosphorylates the pump, which causes it to change shape. What it does is essentially it's open towards the inside of the cell, it has those sodium, and then it gets phosphorylated, and ATP comes in, acts on it, and so then it closes towards the cell and opens to the outside of the cell. By opening towards the outside of the cell, the 3 sodium are then released, and 2 potassium come on in, and then the binding of that potassium causes the phosphate to pop off, and then it goes back to its original shape. So here we go. It has the 3 in there. It goes, the sodium come out, the potassium come in, the phosphate pops off, and it goes back to being open towards the inside, and the potassium come on into the cell. So that's the main question that you're going to get asked. 3 sodium out, 2 potassium in. That's the question that you absolutely love. They love the question that it uses ATP, so it's active transport. And they love the fact that it is what restores and or maintains the resting membrane potential. Those are the big test questions you're going to see when it comes to sodium, potassium, ATPase. Sweet. So continuing more along the lines of these nerve impulses, now that we've gotten a lot of the info out of the way, we have the nerve signals are sent, I should say they're started and sent, as well as travel through a process called depolarization. This is where the membrane potential of a cell becomes less negative compared to its resting state by ions moving across the membrane. So ions are going to move either in or out in order to cause, in this case specifically, ions move in order to make it more positive to begin the process of sending the message. And it does this thanks to those ion channels that we call gated channels. Gated channels, think of it like a gated community. They kind of have a security guard right there, and you have to meet specific conditions in order to enter the gated community. So some very specific things have to happen in order for that gate to open, for the things to go through. We have the ligand gated and voltage gated. So our ligands, or chemical gated, Ligands are chemical messengers that are going to bind to a receptor site located on that ligand-gated ion channel, which causes it to open. Ligands are often, at least specifically when we're talking about neurons, they are neurotransmitters such as acetylcholine, epinephrine, glutamate, glycine, dopamine, serotonin, etc., etc. There's a bunch of different neurotransmitters. The main ones we talk about, acetylcholine, this guy right there, he is going to be your best friend for all of physiology and beyond. You talk about acetylcholine through most of the program, especially if you go down the neuro route like I did. So, and ligand-gated channels, these usually initiate the signal. So the ligand, typically the neurotransmitter, comes and binds onto the receptor, causes it to open, so that way we can get the ions to move in and out. These are more located on the dendrites as well as the cell body, right in this whole portion right here. The dendrites and the cell body are for receiving messages, for starting signals, and then the axon is where we do a lot of the propagating, or like the sending of those signals off to other parts of the body. Those are our ligand channels. Next, we have voltage-gated. So these voltage-gated ion channels have special sensors that monitor the membrane potential voltage. So it's looking and it's checking the difference in voltage from outside of the cell compared to inside of the cell. And when it reaches a specific voltage that we call the threshold, they will open. And then the ions can come in or they can go out of the cell. The voltage-gated channels are made to propagate or spread the signal down the axon of the cell, of the nerve cell. Cool? Everything good so far? Keep on going. If at any point you do have questions, comment them down below, or a very quick Google search. Start learning now how to use Google effectively. The more effective that you are at Googling and finding information, Googling, Binging, whatever you want to use, the more effective you are at finding information, the better off you're going to be in this program. That is the number one thing that you can do, is learn how to find information and how to find it quickly. So the membrane potential, ways that the membrane potential can change. Membrane potential changes when either ions cross the membrane. I should say concentrations of ions across the membrane change. So all of a sudden we have less sodium or less sodium on the outside and more on the inside, or less potassium on the inside and more potassium on the outside. And kind of hand in hand with that, membrane permeability to ions change. So, for example, permeability is talking about how easy it is for them to come across. So, for example, if a sodium channel opens, sodium will rush from the outside of the cell to the inside of the cell. This would say we are increasing permeability to sodium. We can also say if potassium channel opens, potassium will rush from the inside of the cell to the outside of the cell. This would also be called increasing permeability to potassium. It doesn't necessarily matter which direction the ion is flowing. It just matters how easily and how quickly it can get across the membrane. That's what we're talking about when we talk about membrane permeability. This is more of a topic that you're going to touch more on in Physiology 1 than here in Cells and Tissues, but it's a good way to introduce it right now. So there are two types of signals that we talk about when we talk about messing with membrane potential. We have graded potentials and we have action potentials. Graded potentials, these ones can vary in size based on the strength of the stimulus, which is why we call them graded. If they are not strong enough, they will not be passed on as a message. They can be a smaller stimulus, and it creates a little local depolarization, a little local thing happens, but it does not, it dissipates before it really gets to where it would need to go. And these ones are typically ligand gated or mechanically gated channels. Most like ligand is the one that we'll focus on here. So these are our graded potentials, and graded potentials can be excitatory or they can also be inhibitory. They can make it easier for us to send signals, or they can actually make it more difficult for us to send signals. You'll talk more about that in physiology. I just wanted to lay a little bit of groundwork right there. Action potentials. These are what the graded potentials can become. If the graded potential is big enough to reach a specific point called threshold, they will turn into action potentials. These are large, rapid, and all or nothing electrical signals, meaning that either you get an action potential or you don't. And all of the action potentials are always the same size, no matter how strong the stimulus is, as long as you reach the threshold. So these are all or nothing. So either they're happening or they're not. If a graded potential, so let's say our threshold right here is at negative 50 millivolts, it can reach negative 50.0003 or 1 millivolts. But if it doesn't reach negative 50, an action potential will not happen. But as soon as it hits that threshold, boom, an action potential is going to set off, and we're going to get a signal sent all the way through the axon. Couple little details. It's about a 20 millivolt depolarization to reach threshold. And the threshold is reached thanks to those ligand-gated ion channels, again, because that is part of our graded potentials. So graded potentials can turn into action potentials. And these ones involve our voltage-gated sodium and potassium channels. So this is what those action potentials look like. We have some of our graded potentials from our ligand-gated channels. So this one's kind of weak, and then it comes back down to resting, and then you get a little bit of stronger one, and then it comes back down to resting, and then you finally get one that reaches threshold, and boom, we get that all or nothing response right up there. The phases of our action potentials, we have 4 phases that we look at. There's a resting state. So this is when the cell is just waiting for something to happen. Depolarizing phase, where we're getting more positive. Repolarizing phase, where we're getting more negative. We have a hyperpolarizing phase, and then we get back to our resting potential. The resting potential, this is all of the important information that we need to know right here. So the ion channels are closed at resting potentials. I should say the gated ion channels are closed. The cell is at resting membrane potential, and the sodium-potassium ATPase is keeping it there. A stimulus causes either the mechanical or the ligand-gated sodium channels to open, and if it if the cell depolarizes to threshold, so if the cell gets positive enough to threshold, voltage-gated sodium channels open, and sodium rushes into the cell. The cell becomes positive. After we get the full depolarizing, we get all the way up to the top. When we reach around 30 to 35 millivolts, those voltage-gated sodium channels close, and the voltage-gated potassium channels open. Potassium is going to rush out of the cell, making it, again, more negative. Potassium channels, they start to close right about around between threshold and resting potential. They start to close, but they close very slowly. So potassium is still leaking out, and we end up overshooting our resting membrane potential, where that is, and that's called hyperpolarization. And then our good old sodium-potassium ATPase is going to restore it back to resting membrane potential and then maintains it there. So way back, this is what I was talking about when it says it restores it to resting membrane potential as well as maintains resting membrane potential. This sort of step by step, what each of these phases means, what channels open versus close, all that kind of stuff. This is very, very highly tested material, both in this course as well as Physiology 1. So we really want to remember all of this good stuff. So all of that was about, making that first action potential. Propagating is referring to now the spreading, this continuing of that signal. And when we look at this, all of these ion channels, you can kind of think of it like this. Actually, I'll just use this. Where these ion channels that we saw in those previous pictures, those ion channels are located right here. So we get the sodium and potassium, sodium channels open, we depolarize, and then the sodium channels close, and then the potassium channels open, and we repolarize. That's all happening just in this little tiny chunk of the membrane. But what happens is when sodium comes in right here, it's going to dissipate a little bit and carry over, and that's how now then the next spot, right here, this whole spot, has those same channels, and suddenly it creeps up, and it hits threshold, and that chain reaction goes off. And then more sodium comes in, potassium goes out, some of the sodium spreads across, and now this region does the same thing. And that's how these signals propagate. It happens incredibly fast, but that is how all of hit the wrong button. That is how all of this works. That's how they propagate, and they're going to propagate down the entire axon. Let me double check something really quick. If I'm not mistaken, yeah, way back in lecture number, it was week 6 of this course, we talked about the nervous tissue. So this whole process is happening with that nervous tissue. And remember when we talked about those myelinated sheaths and how, they had pieces of myelin covering chunks of it like this, and how it speeds up the time that it takes for signals to travel? This is exactly why. So rather than having to send this signal and have all of these ion channels through the entire membrane work, these ion channels here do their job, and then sodium comes in and it dissipates and it spreads over here, and then all of a sudden, boom, this space goes through an action potential, and the sodium travels. So it limits the amount of action potentials that we have to perform and how many how many channels and everything are involved, how much stuff actually has to work in order for the signal to progress. So that was the importance of those myelin sheaths and those nodes of RON-VA. Sweet. So that is kind of the lowdown on our propagation. Now, we also want to make sure that we space out our action potential signals so that we don't get overlapping information. This is something that can happen with stuff like the muscles, especially if we get too many action potentials, just like action potentials shooting off one after another, it can cause our muscles to contract, and we can actually they can contract uncontrollably, and that's something that's called tetany. I think we've all heard of tetanus shot. Tetanus is from a specific bacterium. I don't remember the scientific name for it off the top of my head right now. The Clostridium tetani. Tetany is uncontrolled muscle contractions, where literally that bacteria actually messes with this whole refractory period and this whole thing, and actually forces our muscles to aggressively contract and can cause some serious damage. So that's why we have this refractory period. So we prevent that from happening normally. There's two types. There's an absolute refractory period and then a relative refractory period. The absolute is during this time you 100% cannot have another action potential generated. It starts as soon as we hit threshold and continues until we get back to resting membrane potential. And this is actually correlated to our voltage-gated sodium channels that control that depolarization, because I hear about negative 55 at threshold. At threshold is when they open, and then they close up here at the top. They don't reset until they get back down under threshold. So the voltage-gated sodium channels are not even available to be able to send another signal, to be able to create another action potential, until they fully reset all the way down here. So nothing is going to happen here in the absolute refractory period window. The relative refractory period means that there can be an action potential that happens, but you need an exceptionally strong stimulus because you're extra negative. Now, instead of going from negative 70 to negative 50 or negative 55, you have to go from negative 80 to negative 55. So you need a much larger stimulus to send that extra signal. Okay. This last slide, I went ahead and made this for you guys. This is going to be, and I actually I can show you I have my paper version right here. I made way back in Try 2. And this whole process of understanding where specific things open, what's happening during each part, where things open, where things close, when are things more permeable, those are all the kind of test type questions that professors really, really like to ask. Refractory period. There we go. So I would recommend this specific thing right here. Either draw your own wave just like this and write all of this out on a piece of paper. That's what I would recommend. I think it helps you learn it a little bit better. But do something like this and keep this. This is going to come in handy a lot in the near future. And that is our full lesson on the neuron function. Thank you everybody so much for watching. Of course, have a wonderful day and good luck studying.